Pat, I don't know if you want to try, are you going to be sharing your screen, right?
Oh, yes, I am. Should I check that?
Just maybe give it a shot now.
Okay.
Great.
Okay. So when you record these, do you post them somewhere or what? How does that work?
I'll give you
I had a few people ask me about it, if they could see the recording.
I'll give you the file and you're welcome to share it, with whoever asked for it.
Okay, great.
Yeah.
Thank you.
I'll put it on like a link on the Microsoft 365, the cloud platform, and you can just send the link around.
Oh, great. Thank you very much.
Sure. Okay, so we're at 1:15, and this is a talk, a monthly talk that we have in the Department of History and Anthropology about a work in progress on campus at Monmouth University. And today we have Pat Cresson, who will be talking about her artwork, and I think her students' artwork, inspired by marine life. And so I'll hand it over to Pat.
Okay. Hi, everybody. Thank you so much for coming. I'm excited to show a portion of the work that I did for this project and also my wonderful students' work. So I'm going to share the screen and start. All these windows. Okay, so this is work that was done as a result of a faculty enrichment grant from the Urban Coast Institute for fall 2020 and thereafter. And the title is The Interface Between Marine Biology and Creative Microscopic Inhabitants of the Sea, Drawings and Works of Art. And I would like to give a special thanks to the Urban Coastal Institute, if any of them are present, Tony McDonald, the director, Tom Harrington, associate director, Carl Villacobo, communications director, Aliyah Satku, administration assistant. They've all been wonderful. And also a special thanks and shout out to the Department of History and Anthropology and Geoff Fouad, and they're allowing me to give this presentation. So I thought, I wanted to talk a little bit about the link between art and science. Art and science are very similar in some perspectives. Both science and art are human attempts to understand and describe the world around us. The subjects and methods have different traditions and the intended audiences are different, but I think the motivations and goals are fundamentally the same. So these were two quotes that I thought fit what I was showing today. Art does not reproduce the visible, rather it makes visible. That's Paul Klee. There is nothing, sir, too little for so little a creature as man. It is by studying little things that we attain the great art of having as little misery and as much happiness as possible. Samuel Johnson. So this project started with it's first branched off from looking at COVID-19 models. But it also, at the same time, I was reading this book, Colliding Worlds, How Cutting Edge Science is Redefining Contemporary Art. When artists and science get together, creative ideas are generated. Collaborative science and art projects are conducted more and more, and one obvious benefit is the greater visibility of the research through the artist's work. The scientist can act as a conduit of research for the artist. This adds another layer of meaning to the artwork and is an entry point to conversations around models, marine biology, protozoa, and microscopic life in the ocean. But what's in it for the scientist? It reinvigorates a curiosity about the material and brings an outsider perspective, but one that is expert in observing, and that would be the artist. So although the COVID-19 virus has proven itself to be a horrible plague, I thought that the models that they were showing on cable news were actually aesthetically beautiful. So I started studying and collecting pictures of them and thinking about their evolution and aesthetic beauty. I then began to study other virus scientific models and found other equally impressive visual illustrations for deadly viruses. These images were all in the public domain from laboratories and scientific studies, such as the Center for Disease Control. And here is an image of the Zika virus. And I thought that was an interesting segue. This is an image, this is a cross-section of a metacy plankton, which we know as jellyfish, and of which there are 200 species. And this was found off the Challenger expedition at 600 fathoms off the Strait of Gibraltar. I thought it was interesting to see the correlation between the two, the same sort of geometric shape, the interesting patterns, and then the sort of, dynamic symmetry that you find really in all aspects of nature. The benefit of artists to science can be elusive, but chief among them is a reminder of the importance of open-ended exploration. Another is to be asked questions by someone who spent even more time simply looking at the object of scientific inquiry than the scientists themselves. The scientist is reminded of the power of creative curiosity, which is sometimes lost in focused research. The artist also asks questions based on hours of observing the objects firsthand. The artist becomes a documenter or illustrator of the work in a way that is not usually perceived in the scientific community. So this is the book that was a great deal of my inspiration, Art Forms from the Abyss. And these are drawings done by Ernest Haeckel based on the Challenger expedition, which was actually the first oceanographic expedition. And by the way, if there are any scientists listening and I make science boo-boos, please feel free to correct me at the end. But the reason for the word abyss, the bulk of the oceans are in darkness, thus the name abyss. My artwork was partially inspired by drawings from this book and the Challenger expedition. This is a drawing from 1828. It's a cartoon of the times showing monster soup. So it's basically showing what they found in the water. Microscopes, they began looking through microscopes in the mid 1600s. And so they were really floored to see that there were so many different living organisms in a drop of water, and calling it monster soup. This is a picture of Ernest Haeckel, and he was a great German biologist. He was one of the scientists of the 19th century who was changing the worldview of organisms. His detailed drawings and studies, particularly from the HMS Challenger, which was 1872 to 76, circumnavigated the world. It's interesting to note that that first expedition gathered 13,000 specimens and recorded over 4,000 new species. Later, detailed drawings were done by Haeckel, who didn't go on the expedition, but from actual biological specimens. And then they were perfected by his lithographer, Adolf Gilch. Here's an 1885 woodcut image of the Challenger's voyage, the map of the voyage. They started with 233 men and 6 scientists and academics. The expedition ended with only 144 men, 7 having died, 23 invalided and dropped ashore. And it's fun to note the rest jumped ship at Cape Town. It was a very difficult journey, and they spent 713 days at sea, traveled 130,000 kilometers, and as I've said before, collected 13,000 specimens. So this is one of the drawings that Haeckel did, and you can see how beautiful they are. They're very detailed. Let me see here. Yeah, they're very detailed, they're very elegant. And his drawings, although based on science, were intuitive and stylish. Observation and representation are evident in both art and science, but how and what one sees can differ. Haeckel's apologists have said that he only showed you what you wanted to see, more of a norm in art. The mark of a great scientist and bold artist is that they consistently question the world around them. So these are some of the other, these are jellyfish. These are some of the other drawings by Haeckel from that book. This is a cross section of a jellyfish. And this I think is so beautiful. The rendered views of seashells. Adolphe Philippe Milot was a French painter, lithographer, and entomologist. He illustrated many of the natural history sections of Petit Larousse and was the senior illustrator at the Museum of Natural History. He was also a member of the Salon des Artistes Français. And I found his species charts and posters to be very inspirational. And I did also use those as inspiration for my drawings. This is a 1907 scientific illustration of a seahorse. And I'm just amazed at how beautiful, seahorses are. I first saw seahorses at the Baltimore Aquarium years ago, an exhibit just dedicated to seahorses, and I've been enthralled with them ever since. Drawing requires intense concentration on the subject or specimen at the time working, not called for necessarily to that extreme when doing photographic illustration. And so potentially it provides an understanding different than that revealed in photography. So these are just to give you a little headway into my own drawings, are some examples of drawing and how drawing is created. And on the left, you'll see this is basically all of these drawings have been done in black ink on watercolor paper. So you're going to see just pen ink lines on the left. And as they get closer together, they create a more dense aspect. As they're farther apart, they are less heavy. So that if you look at the example on the right, which is early Renaissance cross-hatching of drapery, you can see that where the cross-hatching has been gone over and over, things recede back into deep space, and where there is no lines or white space, it comes forward. So that's how they were creating volumetric shape. And there you can see some more examples of that. This is another accepted technique of drawing, which is stippling. So using a heavier pen, thicker pen, you can get a heavier mark, and then using a lighter, finer point pen, you can get less heavy points. These are the tools that I used for this project. It was a variety of basically they're felt tip pens. There's some Japanese Sakura ink pens. They're in all different widths. You can see some of the examples of the drawn lines on the right on the watercolor paper. And these are the different species that I looked at, in the first set of drawings, and then in the later under the sea pieces. So you can see they go from very small marine protozoa, diatoms, hydras, flagellates, to larger sea pens, sea lilies, comb jellies. And then I started exploring different species of fish and crustaceans, crayfish, lobster, jellyfish, different types of shells. So these were the first drawings that I did, and they were marine protozoa. And I added, you can see the different colors. Those were Japan paper collaged onto the drawing. The way the project developed was that between me spending many hours teaching online for the COVID semester, I was also helping my students work on their senior portfolios. So I ended up working myself, in between online teaching. I've always been inspired by the models of Ernest Hegel, and throughout his life, he made many detailed drawings of microscopic life forms and organisms and one-celled marine life. So without having actual microbes, which maybe I'll get a chance to see at some point in our biology department, I used his drawings and scientific photographs as a jumping off point for my own drawings. So these are examples then of my own drawing. And I've been particularly interested in marine protozoa, diatoms, flagellates, hydras, jellyfish, fish, and corals. And I didn't really know what those were until I started looking into it. So these are moss animals. These grow in colonies in water. This is one of my favorites, and I had to learn a new word for this. So I've been doing single drawings. I've been doing double drawings, which are called diptychs. I've been doing triple drawings, which are triptychs. My students will appreciate this. And I've been doing four-part drawings, which are quadtychs. So this is a quadtych, and I will show you just so you can see maybe a little bit more of the inking, you know, how I created volume in various different ways, overlapping the pen, and just, you know, as again, dense areas recede in space and white areas come forward. So this is a sea lily, and they're related to sea urchins. They're shaped like a lily, and they have a cup-shaped body, feathery radiating arms, and either a stalk or claw-like structure with which they are able to attach themselves to a surface. This is another example of the moss animals. These are, you know what, I get them confused. These are radiolarias, and that was one of the favorites of Haeckel. And it's amazing to me, it's amazing to me to look at the detail and the complexity of these little microscopic organisms. I mean, I just found it absolutely fascinating. And I, you know, they're so beautiful. So I have used poetic license. I have mixed up things. I've reinterpreted them. So they're not scientifically 100% accurate. They're more expressing some of the whimsy and the fun I see in this type of thing. These are flagellates, which again are unicellular plants. And I'll go into detail on those again too. So it's really the, you know, a lot of it's in the detail. I also do big abstract pieces that are not detailed at all, but whoops, shoot, what have I done here? Now I'm getting out of order. Yeah, I wanted to show the detail on one. Let me just try to go in here. So I think that the detail and the intricacy and the patterning, it almost looks like Roman armor, is what makes it so interesting and what I found so fascinating. So, you know, I tried to present it in unusual ways, having one half of the picture go over to the second half. You can see I've had it, you know, carry over. This is another example of a radiolaria, and those are microscopic marine protozoa encased in three-dimensional glass skeletons. These are examples of hydras and jellyfish. This is a sea pen. And again, I just thought that was so beautiful. It's a soft coral, but it almost mimics the look of, I keep flipping around here, sorry. I'm trying to get to zoom in a little bit there. It mimics almost the look of a flower, but this is a form of soft coral. These are radiolarias and comb jellies. And I have to look up again because I get these all mixed up, but comb jellies are marine animals with a gelatinous body bearing rows of cilia for propulsion. So those little things sticking out of them move them around in the water. And I just thought this was, I mean, to me, this piece on the right, it almost looks like these almost look like female sculptures to me inside of the shapes. This is a diptych of anemones, and anemones are, you know, use their toxic poison around the rim of their opening mouth to trap their victims. Here's an octopus, and again, a seahorse which I love so much. And there you can see the detail in the octopus. I don't know what I'm doing here. I'm sorry. There we go. Octopus and a flying guimard fish at the bottom. This is another seahorse with seaweed and an octopus starfish on the bottom of the ocean. This was fun, just showing fish swimming through seaweed. So there's two of these trunk fish, which are toxic scavengers down at the bottom, and then a grass fish at the top and a crustacean, a crab caught there in the seaweed. This is one of my favorites. The top one is a flying guimard fish, very dramatic. And then the big funny banded puffer fish below him. And you can see I've started to introduce single lines to represent movement. It's very difficult. It's fairly easy to sculpt volumetric shape by using hatching and cross hatching, but showing motion was increasingly difficult. So I came up with these techniques of introducing single lines. I don't know if you can see around that top fish, that's a trunk fish. There's a little dotted line to indicate that he's, wagging, he's swishing his tail to move. So always trying to show, here again, more evidence trying to show movement. There's a seahorse, two seahorses, and a hydroid polyp, and I believe that's a grass fish at the bottom, and then coral. So this was the first series I did, and then I decided to go on and try to make them look even more like they were underwater. So I decided to spray paint the paper background and then go in and use a toned epoxy to flow over the paper. So these were, this particular series were first drawn. So the way all of these are done is first I draw them in pencil, then I do an outline in black ink, and then I start going in with different pen points and modeling and forming volumetric shape. At this point, I could add, for instance, the one on the right, you can see I've added some Japan paper, what we know as rice paper, as a collage. These are then glued to a wooden panel, and then epoxy is flowed on top, liquid epoxy. I've been adding some tinted color to the epoxy and then allowing it to dry and cure. So these are all examples of drawings that have been, you can't see the panels in my drawing here, but I will show you one later on where you'll see how it's attached to the panel. So this again, these are hydras, and you can see the little cilia on some of them that allow them to move around in the water. I've added the rice paper to give a sense of water and movement. This is marine protozoa. This was a bit of an experiment. I took some leftover paper on the bottom that had been spray painted and glued it onto the drawing and continued the drawing down over that paper and then added the Japan paper, trying to create a sense of movement and water and organic life. This is the first large piece that I did. This one was 16 by 20 inches. And on this, I decided to try a monochromatic approach. So starting with the paper, spray painting in green, putting in my pencil drawing, and then starting to work in making volumetric shape through the different pen points in the green, then adding the hand tinted Japan paper, which was green. This one, as you can see, was attached to the wooden board. There you can see the way they actually look. And epoxy is flowed over the surface, and then I use wax to rub the wooden edges. This was another one in that series. So again, the beautiful seahorses that I love so much, trying to even more with this monochromatic effect, creating even more of a sense of underwater. I've added some air bubbles to the seahorses, and I hope they do produce air bubbles. I think they do, but that was a guess. And so this particular group of monochromatic pieces, I did a few of them. I love this fern, this seaweed on the right. It's called fern seaweed. And I love it because it has these delicate little needles. And then if I can get it together here on this, seeing, you know, some of the detail work here. One of the hard things to do is to create transparency. So especially when it's not in paint. So this is very different. I also work in paint, but this is very different than working in paint. So trying to create a sense of transparency just with linear line. So creating two different pieces and then going over the area where they overlap. This was a bit of an experiment. I tried two different colors on the crayfish and the seaweed, a brown and blue, and you know, plus there was some unicellular microbes floating around there. These are whimsical sea landscapes, and I'm not necessarily sure they're 100% accurate scientifically because you know, I'm an artist, so I'm inspired by science, but I have to use my own whimsy and my own imagination to create these worlds. So again, I think they're pretty accurate, but maybe a scientist will tell me if I've gotten something wrong. Again, here trying to show that sense of movement from the fish and a lot of detail work. You can see the overlapping, the trying to create transparency, and then the effort to sculpt or volumize this form here by going from very black to white. Another one, seahorse, a lobster on the floor. This one is one of my favorites. That trunkfish on the bottom is a toxic scavenger. They're very ugly fish. And for that reason, I'm attracted to them. You've got that beautiful flying gammard fish at the top, and then this wonderful type of anemone, which is, you know, just magical to me, the way some of these things look. This is a triptych, and triptychs are different from three-part series. Triptychs, the center form is always the most important. There's always more meaning in the center form, and the left and right panels tend to be less important. So again, this is the left panel. It's a good example of showing, you know, how I created overlap. Here in this one, you can really see the transparency of the seaweed, that being able to see through it to the form behind it. So creating a real sense of transparency. And this is the right panel. This is the last one I've done and the largest one. This is 18 by 24. And in this one, I was really going more for an all-out marine effect, a mixture of fish and crustaceans, unicellular microbes, you know, ferns and seaweed. Again, it was created first as a spray painted piece of watercolor paper, then pencil drawing, then outlining the pencil in black ink, and then going back in and sculpting those different shapes. So in preparation for my students' project, this was for advanced digital imaging, I had asked them to do 4 different collages, 2 traditional and 2 digital, that were related to marine life, going from microscopic up to larger macroscopic sea life, and a number of different topics that they were to choose. So this is the way I approach collage. I first set out my containers with different pieces of paper in different colors, almost as if, you know, as if I were using paint. And there you can see the board, which has the paper attached to it, and a simple ink outline, which I started to work with different types of paper. So collage is a pretty old technique. It's basically the use of a lot of different materials such as paper, wrapping paper, photographs, old drawings, newsprint, garbage you find, you know, food labels, whatever you want to use, ripped up and then applied to the surface. So this is partially finished. Here it was complete, and I coated it with a matte media to protect it. because I was going to then cover the surface with epoxy. So there you can see coating that. And in all of these, in this series of little collages, I've added a little surprise. It's like a little treasure. It's a little three-dimensional jewel that's been added. So here you can see where the board has been taped off to form a well, and the liquid epoxy. In this case, I was using clear epoxy. If not, I would put dye into the epoxy and mix it up well, or you can also not mix it up well. I've done that, and have the epoxy appear different colors in different areas. Once it's dried and cured, I pulled the tape off, to reveal the edge of the epoxy, and then that was the finished version. So these are some of the traditional collages I did, and you can see they're very different from the drawings, just utilizing paper to basically painting with paper and trying to get the same sense of marine life as, I had in the drawings. So this one, you can see all of them have that little surprise treasure in them. So now I want to show my wonderful student work, and this is from Advanced Digital Imaging. And these students, this was their first project. So they've gone on to do even more impressive projects since then. But these were the two traditional collages they had to do. And these first two are by Gina DeCaro. And the topics they were given to study, this one is by Hannah Canardi. They were given these topics: to look at the food web, the microbial food web in the sea, to look at symbiotic relationships in the sea. For instance, the barrel sponge, home to symbiotic microbes, to look at the role of light within the ocean, the health of the ocean inhabitants, whale exhalations and healthy respiratory biome, ocean outbreaks, environmental impact, the warming oceans, hydrothermal vent bacteria, and coral reefs, dependence of coral reefs and microbes. This one is done by Laura Cass, and this was showing the more alive, healthy ocean on the left and the more diseased. You can see the whale tail on the right, and I don't know if you can see the turtle in the upper portion of the one on the left. She's used painted bubble wrap. There's ripped up painted paper. There's sandpaper. So these are by Shane Castle, and these are all done. And he's a professional seamstress, so these are done as a sewing option. This is one by Kat Methvessel, and she found this wonderful material that was like a map, and treated it and scrunched it up, and then pulled out pictures of sea life. And this is one of the anglerfish. That appeared to be the most popular topic of all the students, was the anglerfish. You're going to see a lot of anglerfish. But this was a really creative approach. The top two are showing ocean microbes, the bottom showing the more macroscopic fish life, and the two top right and bottom left were done in Dayglow paint, and they were photographed with a black light. So he wanted to show the phosphorescence of the ocean. So those two are supposed to be showing that phosphorescence. This is Kelsey Lorenko, and her again is showing the diseased ocean on the left, where garbage and plastic, tons of garbage and plastic, have hampered sea life and caused a lot of death and decay in the ocean, whereas the ocean on the right is the healthy ocean. You can see a different approach here. This is the beginning of the digital collages, and this is Jenna Capitelli's, her interpretation. And again, these are all creative interpretations of what they learned. They were sent to a website, the Smithsonian Institute website on ocean microbes, and it's a wonderful site, and that's where they were getting a lot of their inspiration. So here you see the famous anglerfish that they all loved, with his little antenna and his little luminescent antenna, which attracts victims under the ocean. And they look like monsters, but I was reading that they usually range from 2 inches to like 12 inches. They look like they're 10 feet long. This is Alexa Caluzzi's interpretation of coral, the coral reef, a healthy coral reef. And then this is her interpretation of the monster anglerfish attracting, I think it's his food source. Maybe one of the scientists will correct me on that. But I loved the way she approached the patterning and the color in this one. This is Gina DeCaro's version of, again, the famous and well-liked anglerfish, and really showing the depths, the abyss, what they call the ocean, the abyss of the ocean, and the little luminescent light on the end of his antenna. And then you can see a sort of luminescent jellyfish floating up there and some microbes down below. Here also by Gina DeCaro, she's showing the importance of light in the ocean, and certain fish, well, these are sharks, but certain fish are attracted more to the upper portions of the ocean where the light shows through, and then certain other bottom crawlers are attracted to the dark, deep aspects of the ocean. This is a piece by Chastity Conception, and it was her interpretation of the ocean as a sanctuary. And I thought that was really interesting. It almost has like a fishbowl effect, but there was sort of a magical quality. This was another one of Chastity's. This is Kelsey Lorenko's beautiful 2 interpretations of coral, showing coral in its marine environment. This is healthy coral, and then this is coral that has been diseased and is bleached. Cameron Ellentuck did this piece with the anglerfish in the top right corner and the pufferfish down below. I love the tentacles. I think they were jellyfish tentacles, and the patterning she got from the background to create this, wonderful underwater scene. And then here she shows the relationships between healthy coral and healthy microorganisms, healthy fish, and they're all happy together there. This is a piece by Laura Casson, and she's showing the Earth heating up. The sphere represents the Earth overheating, and you can see fire coming out of it to represent the overheating, and then the coral being bleached. And the result of that down below is water that's not fully healthy or fully, it's not healthy ocean. And here again, you can see coral. This is Noor Al-Malala and her showing the benefits of whale exhalation and how even dead whales on the bottom of the ocean can produce exhalations that are healthy to our oceans. This is Hannah Canardi's view of what's happened in certain parts of the world on certain beaches. I hope none of them in New Jersey, but the horrible, you go to some beaches and your heart sinks when you see all the garbage and all the, desecration of our natural, beautiful ocean. And then this is her interpretation of the bright side of things, where the ocean is healthy and the creatures in it are healthy. And I love the sort of magical fairy tale quality of this. This is Selah Prater's, serious look at ocean outbreaks, how pollution and disease can really damage our waters. And this is a beautiful illustration by Chris Williams. I love the sort of symbolism. It sort of goes back to the old maps and diagrams and all the different, the lush life of the ocean. And there he closes with images from my favorite biologist, Ernest Haeckel. So that is the end of my presentation. I don't know how I did. Yeah, I guess I did pretty good on time. So I was wondering if anybody has any questions or comments they want to make.
Pat, if I could jump in, Fred, I was fascinated. Thank you for pitching it to non-artists, and I like how you showed us some of the techniques that go into building a work of art, like the hatching and the color, and also the way you experimented trying out how to achieve an effect. I thought what was interesting about the project is that you have an inspiration and then you carry it through over a long period of time, focusing on that, and then apply it to the teaching. And I was just wondering, at the end of it, what changed in you? That is either artistically or as far as understanding. It's interesting that I guess the students' work seemed much more environmental. And it seems what you said anyway, that your interest was primarily aesthetic, the beauty of these creatures. So I was just sort of wondering, as far as an artistic and just sort of personal trajectory, what happened from the beginning into the end, would you say?
Okay, that's a great question. And I mean, I do think some of the students' work is aesthetic as well, but I've found in my teaching that students work best if they have specific problems to solve. So that's why I gave them the 6 topics related to the environment and the ocean. And as opposed to just sort of make something beautiful, although some of their, many of their things are beautiful. How I changed, I have been a lover of the ocean for years, all my life, even from the time I was a small child. It's just the COVID thing. I was trying to find something positive in the COVID thing, and just starting to look at the models, I was like, these are really magnificent. How can how can they be these pathological killers? And that led me on to looking at Ernest Haeckel's biological, drawings. And then this is the way I work in art. You start at a point 1 and you just move on to point 2, point 3. You just open your mind and you keep letting your imagination take you from one thing to the other. So I would say that one thing that I've come out of from this is a more intense interest in science. And, I want to look more at other aspects of science. I mean, I was just, amazed at some of those unicellular microbes, the flagellates, and with their little armor, intense design. They were just fascinating to me. Does that answer it, would you say?
Yeah, I mean, each journey is an individual one.
Yeah, but thank you for your observations. They were interesting. Anybody else have any questions they'd like to ask or comments? Students can also say something if they're there.
Hi, Pat. This is Jason Adolph.
Hi, Jason. How are you?
I'm good. How are you?
I'm looking forward to looking through his microscope.
Yeah, there's so much I want to say about your presentation. Thank you. Beautiful work of yours and the students. One thing struck me, the very first quote that you showed, that I forget how it went exactly. Art doesn't reproduce the visible, but makes something visible.
Correct, Paul Klee. Yes.
I just want to emphasize, as a person who spent a lot of the last 20 or so years looking down microscopes at single-celled protists, we don't see under the microscope what you're portraying exactly, because of issues of depth of resolution and depth of field, and it's just real life. It's hard to see, and as a scientist, I've learned to depend on artists' depictions of what we're looking at to actually know what it looks like, because for instance, a single-celled organism, you would have to take pictures of it from many different depths of field to get the picture of the entire thing. Right. And you don't see that all at once in the microscope, unless you're using an electron microscope, which we don't all have handy all the time. So I just want to say how much I appreciate, the work that you're doing, and yes, invite you to come and look at some of the local phytoplankton that we find. They're beautiful. I try to impress it on my students all the time, and I think what you're doing does a much better job of doing that than what I try to do with the microscope.
Oh yeah, well that's really interesting. And the images you sent me, I know what you're talking about. They're very flat. And that is kind of how I started my drawings. They start as just outlines, and I'm kind of making up, the volumetric part, not totally, but yeah, I'm sure it's not 100% scientifically accurate.
Well,
But
and there's something else you said, that you referred to artists as experts in observing. And scientists consider themselves experts in observing as well, but in a different way than what you're talking about.
correct, right,
And
yeah.
I think it's a great coupling, a great marriage between art and science when you're talking about trying to understand what microbes look like, because we can't see them without the aid of something.
Right. Yeah.
And
And I think, yeah, and science can be inspirational to artists, and artists can be inspirational to scientists. Yeah. That's very interesting what you're saying. Thank you. Thank you for those observations as a scientist. Did I make any science boo-boos?
no, you're pausing on recalling radiolarians at one point, but then you got it.
I had to actually study my pictures to learn these names.
Can I ask just a sort of one bridging the two, that do you ever think, Pat or Jason, about the sort of scientific function of beauty? I heard a program a while ago where a scientist, a biologist, and she said she came to graduate school because she wanted to know why two flowers, the aster and the other one that grows right next to it, looked so beautiful together.
Huh.
And her advisor, the people in the biology, said, well, maybe you should go into art. But she stuck with it, and indeed found that there is a function in attracting insects, I think it was, that the reason why their beauteous appearance together made biological sense, so that might be a way in which, biology and art can teach each other something.
Yeah, that's a great point.
Yeah.
Yeah, and it's all connected, It's really all connected.
Maybe the world isn't beautiful for nothing, you know what I mean?
Anyone else? I want to see if these students, are there students here? A few of them, yeah. Did you guys like seeing your images, students? I should say that this is their first project. This is advanced digital imaging. They had learned Photoshop before this, and they were asked for these digital collages to combine all kinds of information, their original photography, stock photography, drawings, diagrams, maps, whatever they could find to express their point of view. And now they're on their second project, and I feel like they've even advanced way beyond this first project. So they're moving along great.
Both artistically and as far as environmental consciousness goes, the sort of social engagement of art is something that I think is so inspiring, especially among young people,
Right, yes, yeah. I mean, you make me feel a little guilty there, Fred, because, unlike my colleague, Kimberly Callis, who I think is extremely focused on, conservation and the environment. I mean, I am in my own way because I just appreciate it and love it, but I've never been particularly political in my work. And as you say, more interested in the beauty or the underlying structure of, what I'm looking at. But maybe this will lead to new things.
well, guilt provocation wasn't my point, my purpose, but just the fact that you're bringing out the breathtaking beauty of these creatures inspires surely everyone to want to make sure that they stick around. So.
Pat, you asked if the students enjoyed seeing their work. I certainly enjoyed seeing it. And it was not surprising to me, the level of quality, because I've been in the gallery and I've been I've seen Monmouth student work displayed elsewhere on campus many times. So I'm certainly expecting, to be blown away, but I always am. And it's what I'm wondering about students on the call is Professor Cresson talked about her march to this inspiration from one image to then playing around with a variety of techniques to get at what was tugging on her imagination. But this comes to you as a theme that's assigned, and yet I find the creativity just really sort of equally explosive. And I wonder, is it easier in a way to be given somebody else's theme or inspiration and set loose to run on it, or is it harder?
Okay, guys, give them an answer.
I personally think it can be kind of limiting, but also in a way it forces you to think more creatively, because you're given a set of specifications and you have to hit all those points. But with COVID, I know it's harder to get some materials that we would normally have access to. So it kind of forces you to see the things that you use every day in a different light. So I don't know.
That's a great point, Kat, because these students were restricted. Normally, they can go out and get any materials. We have large format printers. They print off beautiful, 16 by 22 color images of what they do. So they were restricted. And I think that's a really good point, Kat, that you guys pushed yourself even with the restrictions that you had. Any other students? Did you can get back to me, guys. Were the topics I set out for you restrictive or helpful?
I found them a little restrictive.
Okay.
Wait, did you say okay?
Okay, so that's good. So you'll give me feedback on how to improve.
I think conceptually, sorry, my bad.
Okay.
Go ahead, Kat.
I was just gonna say, I think conceptually, some of the topics were a little broad. So the role of light in the ocean, it could pertain to how it helps organisms grow, or how it harms organisms. Like with the deep sea, there's not a lot of light down there, so you can really play with the contrast of it. But I think it's so broad, it can be kind of hard narrowing down a specific topic that you want to follow with it.
Okay, all good points.
I don't see restrictive also as a negative necessarily. It can be a value neutral term, because often restrictions, I think, unleash great combinations mentally. I think about my daughter who writes. Sometimes when she's given a prompt, it's like permission to play in another sandbox, where she's less responsible for the genesis of the piece, and can kind of abandon herself to take approaches she might not have chosen for her own inspiration. And it, semi-related, I think of how somebody like George Lucas, was like a better filmmaker with $1 million than $20 million. So, or however many million he actually has. So I even sometimes watching the kind of the shows that my kids like about people doing creative things and then getting criticized by a panel of mean people. You see how sometimes some restriction that they're put under really works great for them.
Yeah, those are great points. I found in teaching for the past 30 years or whatever it's been, that some restriction works better than no restriction. Being a sort of loosey-goosey artist in the beginning, there weren't as many restrictions, and I found that students were just kind of like, well, Pat, what do you want us to do? And no, it's not about what I want you to do. It's what you come up with. So it seems to me, although I'm always open to getting student feedback, that restriction is actually helpful. But maybe what Kat is saying is the restrictions were even too broad for her.
Yeah, I mean, I wouldn't actually assign like a negative or positive connotation to it. It's more of like an observation, I think.
Okay.
Sorry. I know that wasn't exactly what you're looking for, but
No, it's fine. It's fine. And you should see her new series that she's doing. It's very creative. So their second project is much less restrictive. And, you know, I'm sure I'll get equally as good results from that too. Any other questions or comments? Well, thank you very much. I really want to thank the history department for the opportunity to present my work. I sometimes feel like we're all buried in our little nooks and crannies throughout campus, and we can't really, I love this kind of exchange where, you know, we're interdepartmental and we're talking about different topics and sharing. So, you know, I think I've made a couple of new friends in the Urban Coast Institute and over in biology, and, you know, so that's great in my book. I have worked with the science department before with student projects, doing some of their logos over there for Professor Danishkar. And I know my colleague Karen Bright has also done work, and Kim Callis has done work. So I think it's great when we can all share and, you know, interact together. So thank you, everybody.
Thank you.
Thanks, everyone. Thanks, Pat.
Thank you. Yeah, thank you.
Congratulations. Awesome.
Thank you.
Thank you, Professor.
Okay, see you guys.
Thanks, Professor
Bye.
Fouad, for setting us up.
Yes,
Yeah,
thank you.
yeah, he does it. Yeah, yay, Jeff.